4 is a microscope. Now, if we try to look into the definition of microscope into Google and into dictionary, it gives you a lot of definition. But simply, microscope is a tool. Why is it that it is a tool? Because microscope is an instrument used to see. Used to see what? Objects which are living and non-living things by nature that are very tiny, which cannot be seen by the naked eye. Like, for example, organisms like bacteria, viruses, and parasital ova. And what do we need to see into the microscope? We need to look into the details. Details like, for example, structures, shape of these organisms. So I have here with me two types of microscope. We have the mononocular and the binocular microscope. Their difference is the number of ocular lenses or the eyepiece. Mononocular contain one and the binocular contain two eyepiece or lenses. Now for the parts of microscope, first the eyepiece. Now the eyepiece is actually adjustable. You can use any magnification if you want, or you can change the magnification. Now for the mononocular here, we do have 10 times or 10x, meaning that it can magnify up to 10 times. For the binocular here, we do have 100x, which means that it can magnify up to 100x. The next part is the draw tube, or sometimes known as the body tube. Now the draw tube and body tube holds the eyepiece into its position. Now the next part is the revolving or rotating nosepiece. The revolving or rotating nosepiece holds the different objectives. Now, we do have four types of objectives. We do have the scanner, low power objectives, high power objectives, and the oil immersion objectives. Now, in order for us to differentiate these objectives, we can see the bond into the different objectives. Now, if we can see red bond, it simply means that that is the scanner. If we can see yellow band, that is the low power objective. If we can see the blue band, that is the high power objective. Now, for a binocular microscope, it contains four objectives. And what is the missing one? We do have the white band, which is the oil immersion objectives. Take note that if we are using the oil immersion objectives, we need to use cedar wood oil or simply oil itself. Now the next part is the stage. Now looking into the stage itself, the stage actually has a hole at the center. Now what is the purpose of the hole? The hole is actually the bridge, or it is the passage of the light that came from the light source, which is being regulated by the diaphragm. Now into the stage itself, we do have the stage clip. Now the stage clip holds the specimen into its position. Now for the binocular microscope here, we do have the stage adjustment knob, which you can adjust the stage clip. The next one is the adjustment knob. Now we do have two types of adjustment knob. We do have the fine adjustment and the coarse adjustment. Now for the mononocular here, this is the coarse adjustment and this is the fine adjustment. What is the function or the purpose of the adjustment knob? It gives, or it focuses the specimen, or it gives a focused image of the specimen. But for our coarse adjustment, looking into its way or movement, it's the up and down movement for the coarse adjustment, and so with the binocular. And for the fine adjustment here, these are the fine adjustments. Okay, it actually gives a finer view of the specimen or the image itself. Now, below the stage, we do have here the iris diaphragm. Now for the iris diaphragm here, this is the iris diaphragm, which contains the knob itself. It regulates the light that passed through the stage that came from the light source. Okay, it is actually adjustable. And we also have here the light source. Okay, for the light source of our binocular microscope, we have here. Okay, and it came from electricity. And for our mononocular, it's the mirror. The mirror serves as the light source of the mononocular. And we also have here the base. The base actually holds the totality of the microscope. For our binocular microscope, we also have here the power switch, power off and on. And that's for the function and the parts of the microscope.